Roberts.
My questions are to the TGA, and these questions go to the approval for Molnupiravir. This was a drug developed in 2014 to treat encephalitis and was then repurposed for influenza but was discontinued after concerns it was mutagenic. Merck then bought the company and used their influence with regulators, such as the TGA apparently, that's to have the product approved as a treatment for COVID on the back of a single trial where the preliminary results supported the application, but the final results showed, if anything, it had negative efficacy. Given the weight of evidence in study after study that Molnupiravir has 0 to negative efficacy, why is it still approved?
Thank you for the question, Senator. Just while one of our medical officers, Dr. Kay Robertson, comes to the table to respond, I would just highlight a couple of things. I take the comment that you made around that the drug company used its influence on the TGA. There is a process that we follow, obviously, in evaluation of all medication. Sponsors bring them for evaluation of safety, quality and efficacy, and that's the process that was undertaken rather than one of influence. I think it's probably important to note that. In terms of the issues that you raise, or the question that you raise around why the medication is still approved for the indication that it has, I'll ask Dr. Kay Robertson to respond to that.
Kay Robertson, Medical Officer, Medicines Regulation Division. Thank you for your question. The TGA considered the evidence to support the approval of Molnupiravir from the dossier that was submitted by the sponsor in accordance with our standard processes, and drew the conclusions that at the time the benefits outweighed the risks. In terms of the specifics of any subsequent information that has been provided to the TGA, I'm actually not in a position to comment with certainty. This is not the area that I work in particularly, and I think that we would be best advised if the Senator pleases to take this question on notice and provide you with further update.
Okay, I appreciate you giving that offer, and I will accept your offer for the question to be answered on notice. But it does surprise me that approval was given on a single trial where the preliminary results supported the application, but the final results showed, if anything, it had negative efficacy. And this weight of evidence in study after study shows that 0 to negative efficacy. So I'm still amazed that it's still approved. The approval required Merck to continue to provide ongoing safety data and testing. One hopes around mutagenicity and interaction with the mRNA vaccines. Have they done that, and does that data justify retaining approval? I have before me the OSPAR that was published that in relation to the studies that assessed the risk of mutagenicity. And we can provide that to you, Senator, in our response. And I'm reading from this, and it says that Molnupiravir and NHC were mutagenic in the bacterial assay with and without metabolic activation. Molnupiravir and NHC were not genotoxic in vitro and in vivo micronuclei tests, and in vivo mutation assay at the C11 locus in the big blue transgenic F344 rats. Equivocal results were obtained in an in vivo peak A mutagenicity assay. Carcinogenicities were not generally required for drugs for short-term clinical use. However, the sponsor has initiated a short-term carcinogenicity study in mice. This was put to the clinicians on the ACM and other invited experts regarding this matter. It was considered that at the time, on balance, that the drug remained to have
a positive benefit-risk balance.
And I would just thank Dr. Robertson for that response, and I'd also just add, Senator, that because you're asking for some very quite specific currency and comprehensiveness of ongoing post-market reporting, we'll take that on notice and bring that information back to you.
Thank you. In 2023, Molnupiravir is top of the pops, Australia's #1 drug, costing taxpayers $654 million last year at $1,125 a prescription. Molnupiravir is 26 times more expensive than the out-of-patent Ivermectin plus Zinc combo, which is about $40 per prescription. And that's what Molnupiravir replaced. Proven, safe and effective. Why are you spending $654 million when $25 million would have done on something that's highly questionable as to its efficacy and its safety?
Yeah, thank you for that question, Senator. I can't speak to the specifics of the amount spent on Molnupiravir, but I can certainly indicate that the second amount that you said would have done. Sorry, I didn't catch the amount. Was it $25?
$25 million for 40. Ivermectin plus Zinc combination is $40 per prescription, and the total for the year would have been $25 million.
Yeah, I think I would just add that I think that the comparison is flawed in that there's no credible, supportable evidence that Ivermectin and Zinc is an effective treatment. So I'm not convinced that you are.
Credible evidence.
I'm not convinced that the comparison is sound, Senator.
So you based a decision on Molnupiravir on one paper, and you're ignoring 100 papers proving Ivermectin success. Does anyone question the process?
I'm going to give Professor Lawler an opportunity to respond to that.
Sorry, Joe. I didn't hear. I didn't hear a question. So I was just.
The question is this. Does anyone question the TGA's processes?
Yes.
For approving
Senator.
drugs? How often do you evaluate them?
Drugs.
Who audits them? Is there an independent auditor?
I'm not sure, Senator, which question you would like me to answer.
All of them.
Okay. Sorry, Chair.
It's okay. Professor Lawler, are you clear on the question placed? So, Senator Roberts, could you please... There's a mixture of questions and assertions moving around here. So let's just step back and Senator Roberts, please place the question.
The question is, how often do you scrutinise your process, and is there an external auditor that does that, who's qualified to do it and to assess the process?
Thank you very much for the question, Senator. The processes that we follow are continually informed by our international collaboration and also by a significant interaction with stakeholders, particularly the advisory committees that we have in respect of the assessments and evaluations that we undertake for products. We also undertake obviously the pre-market review and evaluation of medicines and other therapeutic goods, and we undertake significant post-market surveillance of the goods as well, and we have outlined in significant detail on previous occasions the post-market surveillance that we undertake. I might ask Mr Henderson to add.
Yeah, and Senator, I think you asked about the number of submissions or medicines that we evaluate. So just for context, at the moment there's about 150 applications that the TGA is evaluating for both new medicines plus changes to indications to current medicines.
So what is the point of telling me that?
So I thought that you asked that as well.
No, I didn't ask for the number.
Okay.
Who are your stakeholders? Do they include the sponsors?
Senator, thanks for the question. As a contemporary regulator, we have a broad stable of stakeholders. They include they do include industry. As with any regulator, we work to refine our processes to balance the appropriate observance of safety, quality and efficacy with appropriate access and streamlining processes to bring products to market with a minimum of inappropriate regulatory burden. We undertake annual stakeholder engagement surveys to understand the views of the TGA and the three key stakeholder groups that we survey on an annual basis are industry, health professionals, and obviously it's important that we work with health professionals for a number of ways in that they both inform us and are informed by our decisions. And the third is the community. And it is notable that the responses that we get reflect that the TGA among all groups comes across as a recognised, understood and valued regulator in the Australian healthcare system. We also have other stakeholders with whom we interact. We obviously interact very closely with the state health jurisdictions. This is for a number of reasons. Our decisions on a number of elements, such as scheduling, for example, that we've already discussed today, has significant impact on the state and territory poisons legislations and how they're implemented for the delivery of medicines. We also interact quite closely with expertise across the regulatory sector. We have a number of advisory committees that the membership of which incorporates consumer views, expertise, and also those from the academic and research sectors. And it's also important to note that we obviously have close relationships with our international collaborative regulators. We are part of the International Coalition of Medicines Regulatory Authorities. We're part of the International Medical Device Regulators Forum, and we also work closely with individual regulators such as the MHRA in the UK, the European Medicines Agency and the FDA.
Thank you.
Senator Roberts, I will shortly seek to rotate the call to Senator Rennick, and then I can come back to you. Is this a sensible place to pause?
I'll make it a short one and then you come back to me. Spike proteins can enter the body two ways in the context of COVID, from the virus itself and from the vaccines. What work has the TGA done on the health outcomes of long-term retention of spike proteins by the body after the mRNA vaccines that you recommended? It has been 4 years now, so some good old-fashioned science by the TGA must be available. Is there any assessment?
Thank you for the question, Senator. As has been indicated previously, as with all regulators around the world, we undertake a significant program of post-market surveillance and pharmacovigilance. This includes having a clear and well-communicated preference for adverse events post-vaccine to be reported. Those are reported and entered into our database of adverse event notifications, and along with examination of that and also in collaboration with partner international regulators, we are very much aware and alive to emerging safety signals and act accordingly.
But you haven't done any studies on the retention specifically of the spike protein, because the injections, the COVID injections, dramatically increase the spike protein. So you haven't done any studies of them?
Senator, I'm happy to have any additional response, but what I would highlight is that our role as a regulator is to assess evidence that is brought to us, and we undertake that assessment in the evaluation.
So you don't go looking for it?
If I might, Senator, we utilise that evidence in the assessment and evaluation of products, and we utilise the pharmacovigilance and post-market surveillance exercises that I've highlighted.
Thank you, Professor Lawler.